Ladies and gentlemen, welcome back. For our first session, we have our value sponsor, Thermo Fisher Scientific, who's a world leader in serving science, and they did not stay passive towards the COVID-19 crisis. As a pioneering company providing technology, regions, and data services in life sciences and the research industry, it has already developed many exciting solutions helping actively fight the current crisis. We're very proud to cooperate with Thermo Fisher Scientific, and even more so with today's fantastic speaker, Alain Rico, who is an established speaker in precision medicine, sequencing, and overall R&D innovation enthusiast. In his presentation, Alain will focus on introducing the comprehensive portfolio of genomic solutions Thermo Fisher has developed to fight COVID-19 pandemics. Welcome, Alain.
Thank you very much, Thomas. Not sure I am a fantastic speaker, but at least I have a fantastic French accent, so bear with me for the next 20 minutes or so. Yeah, as Thomas said, I'm from Thermo Fisher Scientific. My name is Alain Rico, and I'm developing the market for precision medicine. In this presentation, I will briefly highlight our very large portfolio of applied biosystem and ion Torrent genetic analysis solution that you can use for your own research on COVID-19. We know that SARS-CoV-2 has rapidly spread around the world, and we all know that it's very important to better understand this disease and how we are susceptible to it, as well as develop vaccine and identify suitable therapies. Thermo Fisher Scientific has a history of response to a public health crisis. We have worked very hard, not just on the SARS-CoV-2, but also during the Ebola, the N1H1, the SARS, and Zika too. And while today I'm going to talk about genetic analysis solution for outbreak research, Thermo Fisher also has a lot of other solutions like personal protection equipment and other scientific research tools like cryogenic electronic microscopy, mass spectrometry, a lot of detection kit, solution for cell and protein analysis, vaccine production, and even clinical trial logistics. Our mission at Thermo Fisher Scientific is to make the world healthier, cleaner, and safer. This is a very powerful mission, and in these days, it resonates a lot. We are the world leader in serving science with industry-leading scale and unmatched depth of capabilities. Researchers around the world, you, are working to gain a better understanding of the novel coronavirus with the goal of rapidly identifying future treatment options or possible vaccine targets. And we know your research cannot wait. So with our comprehensive portfolio of genetic analysis solution, we want to be your trusted partner for all the applications I'm going to discuss today. We have a broad portfolio of instruments, a broad portfolio of platform, and in this slide, you just see an overview of each technology at a very high level. We have our next generation sequencing, the ion Torrent semiconductor sequencing technology. We offer end-to-end workflows to study your viral outbreaks. We have two solutions, both very automated and very cost efficient. The Ion Gene Studio S5 for a cost-efficient and large number of infectious disease research samples. And the Genexus integrated sequencer. With this one, you start from your nucleic acid and you get the report within a day. There's a lot of automation, barely no hands-on time. It's very powerful for your research application. We also have a full suite of genetic analyzer for Sanger sequencing and fragment analysis by capillary electrophoresis. The latest edition, the 6 Studio genetic analyzer, is very convenient for your research because it has this system of all-in-one cartridge with capillaries, polymer, and buffer required for the run, all included. We also have a solution for epidemiological studies. We have a lot of microarrays for that. Both for whole genome genotyping and whole transcriptome analysis. And finally, we are the leader in real-time PCR with over 25 years of innovation. We offer sensitive, specific detection and quantification of nucleic targets. And we have the new Quant Studio 7 Pro, which can help you with his even smarter workflow. I'm not going to spend more time talking about our instrument portfolio because I want to focus and show you how you can use them for your research on COVID-19. We'll review 5 different SARS-CoV-2 research areas and showcase how our solution can help. We will discuss the virus discovery and sequencing, any viral detection, epidemiological surveillance research, vaccine research and development, and host genetics and population genomics studies. Here at Thermo Fisher, we are committed to serving science in the battle against SARS-CoV-2 infections. So let's start with the first application, the sequencing of the virus. For example, to identify new genomes or to generate reference sequences. Whole genome sequencing is a very nice opportunity for your research and for the characterization of microbes. So if you do research about genomic structures, de novo sequencing and assembly of genomes is important for you. And we provide for your project research deep coverage analysis across the genome and very high quality data. Scientists, for example, at the Peter Doherty Institute for Infection and Immunity in Melbourne, Australia, were the first outside of China to sequence that at that time named 2019 nCoV genome. They used the 6-studio genetic analyzer to generate a reference sequence, which is fueling global surveillance research and assay development. We also developed solution for our ion Torrent semiconductor sequencing platform, a technology that, again, really has revolutionized de novo sequencing for microbial research. These systems are, as I said, simple, fast, and most importantly for your research, they have the highest accuracy of variants in the market, the lowest substitution rate for single nucleotide variation, and that's incredibly important for your infectious disease research. We have end-to-end solution, and we also developed, using our flagship AmpliSeq technology, the AmpliSeq SARS-CoV-2 research assay. With this assay, you will be able to sequence more than 99% of the genome of the virus, and it's going to be very accurate. And that's going to be done under a day on one of our genetic ion Torrent sequencer. And again, from just the DNA directly to the report with the integrated Genexus sequencer. Some of our customers very recently also just did FFPE analysis directly for de novo sequencing. It's a very powerful tool. And this panel works on both of the solutions, as I mentioned, and at the same level, even starting from low number of viral copies. Another application of our solution is the detection of the SARS-CoV-2 and the study of the variation in the gene expression in response to the pathogenic invasion. We developed the Applied Biosystem TACMAN 2019 nCoV assay kit. That's a single-plex kit specifically for SARS-CoV-2 detection research. One example of research is infection in domestic animals. Xia and collaborators, for example, use the Quant Studio 5 to study domestic animals, some regular like cats and dogs, and some more bizarre, let's say, like ferrets. And in these ferrets, they actually found that the immunological response is very similar to the one in humans. That means these animals could be used as a potential experimental model for you. You can also confirm qPCR or next-generation sequencing virus detection using our Sanger sequencing platform. Don't forget about that. And of course, you can use our qPCR solution, like the respiratory tract microbia panel we have on the Applied Biosystems real-time PCR machine, to study co-infections or other associated respiratory pathogens. So this is a panel of 42 verified targets, and it's running on different formats, as you can see, and working from different sample type, including nasopharyngeal swabs. Another application is Hamed and collaborators using the qPCR to detect the virus in wastewater and then perform Sanger sequencing to confirm the identity of the virus. This wastewater-based epidemiology approach could help to predict, for example, second waves of SARS-CoV-2 infection. Quite powerful. Obviously, our ion Torrent sequencing platform can also be used to detect the virus. But today, I'm not going to talk about that. I'm going to focus more on other examples of the NGS application in the field of epidemiological surveillance. Epidemiological surveillance is conducted to ensure that the global disease outbreaks match the reference we have and the reference strain, and also to monitor any possible mutation. Indeed, if there's a change in the viral genome, this could impact how the illness will spread, but also impact the treatment options or even the vaccine development. So it's important to understand that. And this is really a major challenge for the virologist, how to predict the pattern of evolution or the emergence of new disease agents. And we know that RNA viruses like the coronaviruses tend to mute a lot. So it was important to study this mutation rate. And using our implicit SARS-CoV-2 research panel I just mentioned, two different teams in Italy found that actually this virus seems to have a relatively low mutation rate. And that was positive news because it means that for vaccine research, the effectiveness of the vaccine could potentially be consistent and possibly over many years. Here's another example of the use of our implicit panel. During a webinar that is still available on our website, Dr. Trish from Children's Hospital of Los Angeles discussed his research and how the SARS-CoV-2, as it spread in the community, undergoes a rapid mutation within host and between individuals. Dr. Trish also showed that these mutations allow for identification of index cases, community spread, contact tracing, but also that this mutation results a lot in amino acid changes that may lead to an antigenic drift with possible negative implication for infectivity, pathogenicity, PCR-based molecular diagnostic, and vaccine development. So you need to look at this mutation using our panel. Last application, famous one, of our sequencing solution is the analysis of transmission between animals. For example, sequencing viruses in potential reservoir hosts. And this led to discovery of a lot of lineages of pangolin coronaviruses, quite similar to the human SARS-CoV-2, suggesting that these pangolins could be considered as a potential intermediate host and basically should be removed from the wet markets to prevent zoonotic transmission. We also offer solutions for host genetics and population genomic studies. So this is a search for potential host contributor to SARS-CoV-2 susceptibility in the hopes of finding genetic markers for COVID-19 progression and biological insight that may point to potential drug targets. This is very important, and we know that to study the disease and how it is impacting the population, how this population is responding to the disease, it's important to be able to stratify. So to do so, for your research, we provide the Axiom genotyping arrays. It can help you for your host response studies and for the risk of susceptibility and the susceptibility. Axiom is a very proven technology which is used in multiple very large cohorts like UK Biobank, FinGen, the Taiwan Precision Medicine Initiative, etc. Collaborators from the UK Biobank, for instance, showed the link between blood pressure and phenotypes and risk for adverse outcomes from SARS-CoV-2 infection. They also found genetic determinants of viral antibody response, and they did that with one of our arrays. Indeed, we have this Axiom design custom genotyping array. It allows you to select more than 800,000 GWAS markers. a lot, like more than 9,000 markers for ACE2, where most of the population array are underrepresented. And you can find the full ClinVar also. So this is the best way to develop your COVID-19 array for research on host response. And that's what we did too. We can help you for your research with our new Axiom Human Genotyping SARS-CoV-2 Research Array. The version 1 of the array is finalized and it's ready for order. And this array includes a lot of the nice features I just mentioned. You can learn more about it next week during a webinar on June 22nd. Check out our website for more information. And meanwhile, please keep coming back to us with suggestions for the next releases of this array. Vaccine research is quite important, and the race basically is on to develop a vaccine for SARS-CoV-2. But to do that, you need to better understand the immune response and how it evolves over time. It will impact your decision about vaccine efficacy, safety, dosage, and also the duration of the protection. This slide is quite busy on purpose, just to show you the complexity and the multidimensional aspect of the immune response. Sequencing the immune repertoire is a very important tool for your research. T and B cells need to be sequenced. The receptor, the repertoire needs to be sequenced. And we have a lot of next generation sequencing panel for you to do so. One example is an experimental plan using Oncomine IGH-LR and TCR-B-LR assays to identify the biomarkers using the comparison of immune repertoire between subjects who develop a mild disease or a severe disease. It's also important in your human response study to look at how the SARS-CoV-2 is impacting some severe associated inflammatory condition. For instance, what's the link with some Kawasaki disease-like or other toxic shock syndrome. So it's important to study in your research using our solution. Finally, the immune response to SARS-CoV-2 is also key to identify any potential antigen drift due to the mutation in the virus genome, because this could impact the spike protein B cell epitope, and it can be population specific. For instance, the 23403A2G variant is quite frequent in some European countries, but almost absent in China. So better understanding the drift in the sequences is key for your research. But when we talk about immunology and immune response, mutations are not the only factor impacting the immune response. The host microbiome also has an influence. And this is known from H1N1, for example, where previous studies using our next generation sequencing showed that. So we know the host microbiome potentially plays a role in the degree of the severity, the hospitalization due to co-infection, and even the antibody response can be varying depending on the gut microbiome profile. So to help you with your research, we developed the Ion Ampli-seq Microbiome Health Research Kit. This is based on our flagship Ampli-seq technology, and this will help you to profile the microbial diversity in your human gut microbiome research samples. We also have 16S metagenomic solution if you want. Another way to look at the immune response is to look at expression of gene involved in this immune response. That's also a good research topic for you. So in the previous slide, I discussed the Axiom array for genotyping, but we also have the Clarium array for gene expression. And you can study host-virus interaction, for instance. In this slide, you have results from 2 studies that suggest that the elevated expression of ACE2 caused by COPD and smoking may explain why these cases have much more severe symptoms and worse outcomes than others. Another tool we provide are the Applied Biosystem TaqMan array with a qPCR panel of 92 genes involved in human response. That's a powerful tool for your research too. I'm gonna skip this one because it's again about Tachman gene expression array. We have a lot of them, and the use of Sanger sequencing for confirming the next generation. I want to use my last minute to remind you that you need a good cell authentication solution for your research when you work on vaccine research, for instance. Indeed, you know that 15 to 20% of the time, the cell line you get from cell repository or from your philo colleagues are either misidentified or showing cross-contamination. So you need to identify your cell line. And we do provide STR solution like the CLA Identifier Plus or the CLA Global Fire Kit based on our 6 Studio for fragment analysis. So do use that. Make sure your results are good. And finally, I realized that 20 minutes are not a lot to discuss the luxury of solution we offer for your research on COVID-19. This was just a brief overview of our instrument and all the solution for pathogen detection, virus sequencing, epidemiological surveillance. I have a very hard time with this word, vaccine research and development, and finally, host genetics and population. So you can learn much more on thermofisher.com/coronavirus or /NGS-coronavirus. And I thank you very much for your attention.
Alain, thank you very much for your insightful talk. And we'll be moving on to our next session, and we'll be bringing up Mr. Holger Muller,